Well, we are on the final stretch, Component 2 mock, Holy Cross mock 1, and we have an essay for 9 marks all about chimps. And there are 3 subspecies of chimps, all with horrendous names, living in West Africa. And we have a new group called the Nigerian group. At least it's got a better name. And what they have done, they say we can use a genetic fingerprinting and so on to look at differences between them, and another technique that you might not know a lot about, DNA hybridization, that we'll talk about. And there's are some results. We got a nice little map, and then we are given 2 possible phylogenetic trees, and these trees show these are going back in time this way to show areas where they had a common ancestor and they diverged into separate subspecies. The actual question itself, what do we actually need to do? How may the chimpanzees evolved into subspecies identified? It should be have evolved, that would make more sense, into the subspecies identified. So that might be our first cloud. So on my answer sheet here, my first kind of cloud might be about evolution. Then Y strands in hybrid DNA separate at lower temperatures than single species DNA. So this is DNA, another cloud here, DNA hybridization. And answering that question. And then using the data and the map to look at this, choose the phylogenetic tree and explain that. So the last little area will be choosing the phylogenetic tree. So those are the sort of areas that we are looking at. Again, I'm not going to write the entire essay. No one's got enough life left in them to watch that. But we'll try and identify some of the key points. And so, in evolution, you studied quite a few examples like salamanders or moths and things like that. So you're kind of looking at natural selection. So things like allopatric, geographical separations, and here you could possibly suggest from this map there might be rivers that might separate them, for example. There's going to be mountains, not really evident from this map, but you could suggest that. So rivers, mountains. So we're trying to use this information that's been given to us, and then just like our salamanders example or moths or whatever, we then go, well, the separated populations have different selection pressures, different mutations. So we go different selection pressures. There'll be variation in those populations due to mutations. So variation due to mutations, and then some will have an advantage over others. So some characteristics will have an advantage in each particular different area. Those will those will survive, reproduce, and pass on those advantageous alleles. Pass on advantageous alleles to the next generation, and then over many generations you get the frequency of certain alleles changing and so on, and the differences accumulate. And eventually, maybe not in this situation here, but eventually you will get new species. So maybe they are unable to interbreed. Here, subspecies, so they probably are able to interbreed, but eventually you might get speciation taking place. So, describing natural selection and evolution, that's your first cloud, that is the first thing that you've been asked to do. So, how may the chimpanzees evolved into the subspecies identified? Next one, looking at DNA hybridization. And why strands in the hybrid DNA separate at lower temperature than single species DNA, and why there are greater differences between less closely related subspecies. Wow. So we have got some DNA. DNA is double-stranded, and we have got bases A, T, C, C, G, and so on, and they're complementary bases, and you can do that sort of thing. Okay. If we heat that up and separate it, then join it onto another separate strand. So, for example, we heat that up, separate it. So, let's take this strand here, A, T, C, C, G, and if we bind that back onto the original strand again, so let's get it back on, so cool it down, and it'll stick together again. Okay. Or we could take that strand CCG, and we could get a complementary strand from another animal, another subspecies, and that might look like T A T G C. And if I use a different colour here, we're going to get hydrogen bonds. Well, hydrogen bonds, there's our original ones. We heated up to break those and separate them, and when we cooled it down, it made our hydrogen bonds again. On this strand, we got that one's okay, TA, that's fine, but hang on, that one isn't going to form our hydrogen bonds between them, whereas these ones are. Okay. So now this is how they are calculating this data. So they're working out the difference, or measuring the difference between how high they need to heat this up to separate this first strand and how high they need to heat the second strand up. And for example, I'm just going to make these numbers up, but say if you had to heat this up to 90 degrees to separate, well, because these two strands aren't bonded together as strongly, then it's not going to require as much high temperature. So that might be 89 degrees C. And you can imagine, if we've got another bit of DNA, ATCG, so that strand's the same, but if we make it even more different, then the only bonds that we're going to make there are these ones that are complementary. We've got two now that don't form hydrogen bonds, and so that might only need to get to 88 degrees C, and that's the data that we've got there. So the question is, why are there greater differences between less closely related subspecies? And so we're explaining the difference between those two, and we're going, if there's different subspecies, there's going to be different bases present on the complementary strand, and so it required different heat levels to separate those. So more closely related, closer to the original temperature. So more closely related, there will only be a small difference in the temperature. So on here, closely related would be these numbers here, kind of 0.6. Bigger difference between these, 1.1. So that's the biggest difference. So that's our DNA hybridization. You just put that into words and diagrams. And then you look at choosing which of these trees we think is most likely. So use the data and the distribution map to see which tree is more likely. So the data, we'll just run through that, and then presumably this map, the areas closer together areas might have more closely related species. So we just need to take some examples from here. So these are horrendous names. So let's get this Nigerian group. There's our Nigerian group anymore. Okay. Let's use a different colour for our Troglodytes. So the one that ends in trog. That's that one. One that ends in trog. That's that one. There, and you can do this on your exam. And that's those. Schweinförderei. 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 And that one. And the last one. Verus. Verus, we'll use green for that one. Verus. There we are. Okay. So we need to go, which is the lowest number, that's going to be our closest together one. So for these trees here, we're looking for which tree has got these two, Trog and Schwein, closest together. So when we look on here, so Trog was in blue, and there we are, there's Trog, and oh no, Trog was in red. Oh no, I've done it wrong. And Schwein, I'll swap the colours around, but it'll be the point will still be made there. So we're looking for which one of these contains more closely related. So tree number one has got these close together. Tree number 2 has got these far apart. Do we want them close together or do we want them far apart? 0.6, we want them close together. So tree number 1 is getting a tick so far. We can do it. We can do it with the furthest away data. So Schwein and Nigerian. So Schwein and Nigerian, we want those to be far apart. So where's our Nigerian group? There's our Nigerian group there, and there's our Nigerian group there. So Schwein and Nigerian, they're far apart on 1, they're not so far apart on 2. So, and we wanted them to be far apart because that's a high number. That gets our tick as well. And we can go into the data for for the others as well, and it kind of agrees with that. When we look at this map, Trog and Schwein, where do we find those? So Trog there, Schwein there, quite close together. And where's our further apart one? Schwein and Nigerian. So Schwein there, Nigerian group separated from them. So that kind of agrees with the mappers there. So choosing the tree, we're going to look at 0.6 for Trog and Schwein. Can't spell that, didn't do German at school. So close on Phylogenetic Tree 1, far on Phylogenetic Tree 2. And so that's what we want there. We can look at this other data, 1.1 difference in temperature for, you could write all these out, I can't be bothered, Schwein and Nigerian. So Schwein and Nigerian, we want them far apart. So far apart on 1, far on Phylogenetic Tree 1, closer on Phylogenetic Tree 2. So again, 1 is what we want there. And then on the map, does the map agree? Yes, it does. So the map has got Trog and Schwein close, which is what we want, and it's got, for example, our farthest away one, 1.1, Schwein and Nigerian far apart on the map, which is agreeing with us as well. So all you need to do is take those data, use the map. So we're using this data and we're putting this argument together, and then you just need to write it out in some sort of sensible order, and make it look attractive, and use the right terminology in English and so on. I hope that wasn't too painful for you.